So hi everyone, I'm Julia, a first year PhD student in the Weinkoff Lab, and I'm working with both Durham University and Magnitude Biosciences. And I'm interested in how automated monitoring of worm movements can be used to screen for neurodegenerative therapies. Current methods of manual paralysis assays can be labor intensive and subjective, and so using the technology developed by Magnitude Biosciences has allowed me to attain non-invasive, reproducible, and quantifiable readouts of worm movement. So I'll just start by briefly explaining how this data is captured and used. Firstly, I pick L4 worms onto 6 centimeter plates on OP50 E. coli lawns with usually around 30 worms per plate, resulting in up to 180 worms per condition. For strains which are not sterile, I also use FUTR to prevent progeny from overwhelming the plate. I then place the plates onto the machine, which is just indicated here by this box, and each plate has a camera to individually monitor it. These individual cameras monitor each plate by taking a video lasting 160 seconds every 5 minutes of the worms. Image processing then takes place through the software to track the worm, where the worm was throughout the monitoring period, and collect information on its speed, direction, and movement. So if I show you what sample data you can get out of this sort of system, you can look here. This is a graph showing a fraction moving. So this is basically the percentage of worms which are moving in the 160 seconds imaging window. And over time, here we have a control in black and 3 concentrations of SMX, a drug which is known to extend C.elegans lifespan. And this is just added to the agar media. As you can see here, the 16 microgram per milliliter condition, it doesn't show as high a peak of fraction moving, so that shows that there are not as many worms moving as in the other conditions at their peak, but at the same time, it does show that there are more worms moving in later life compared to the other 3 conditions. And this is also reflected in another graphical output I use, which is a speed histogram. Here you have between control and the 16 SMX condition. As you can see in the 16 SMX condition, the peak doesn't go as high as it is in control, but there's movement. The worms do move for longer. So from this data, we can say that SMX treated worms don't move faster, but they continue to move for longer. So this system successfully detects alterations in speed and movement, but has been primarily focused on studying age-associated decline. Therefore, I wanted to test whether neurodegenerative strains of C. elegans showed patterns of decline which were identifiable by this technology. And one such result is here. This is something I helped collect. This is the comparison between a control strain and the amyloid beta expressing strain GMC101. It has a full human isoform of amyloid beta driven by the muscle wall UNC55 promoter. So I'll just start this movie here. Then here you can see that this is just a video readouts of the two worm strains, control and GMC101, and you can already see that GMC101 is showing less worm movement. All these white lines are just the tracks. And here in the fraction moving graph, in the light green, you can see control is steadily declining, but then GMC101 stopped at a peak of around 70% of the worms on the plate moving, and then shows a very steady decline, a steady and rapid decline. And here in this bar chart, these are the integration of the spaces underneath these fraction curves and serves as a metric for worm health on these plates. So I've done this with GMC101, as you can see, but I've also carried out this analysis with other models of Alzheimer's, as well as Huntington's and Parkinson's disease. And you can see clearly here that neurodegenerative decline can be modeled by this system. So now that I have a system which can detect early stage movement decline associated with the transgenes containing the aggregating proteins in C. elegans, my next project is to test its efficacy in screening for drugs. And one such candidate is SMX, which is a sulfonamide that dampens folate synthesis in OP50 E. coli, resulting in a lifespan extension for the worms which eat it. It's possible that the lifespan extending mechanism may slow the course of paralysis in these neurodegenerative strains by slowing aging, or it could be through other unknown mechanisms. But what makes SMX especially interesting is that it affects the bacterial folate production with consequences for its host. In humans, we need to ingest folates as we cannot synthesize it ourselves, and there's evidence that our gut microbiomes may be able to create these folates. The microbiome is of growing interest in the field of neurodegeneration, as it may be a source of inflammation through cytokine production or through LPS secretion, and that can exacerbate symptoms of neurodegeneration. Therefore, SMX could potentially act as a microbiome-targeted therapy, and in this model for microbiome interventions. And this is what I plan to be testing in my next project. And hopefully I'll be able to present an update to the story in the future. And so with that, all I have left to say is thanks to both my Magnitude and university teams, and special thanks to Adelaide Raimundo for helping me culture all the worms and helping me with all the assays. And I'm happy to take any questions via Twitter or email, which are on the first slide of this video. Great, thank you very much.